AJ, thanks so much for having me. It's a real pleasure to be here and so exciting to see so many people joining us. I know online webinars can be sometimes a bit of an attention struggle, so I just want you all to know how much I appreciate you all being here. So I'm Amy Willis. I'm the principal investigator of a research group called the Statistical Diversity Lab, and I'll talk a little bit about what my research group does today. We call ourselves the StatDiv Lab for short. I'm also an associate professor in the Department of Biostatistics at the University of Washington here in Seattle in the US. So shout out also to folks who are on the west coast of the US for also whom it's quite early. And today I'll be, as AJ mentioned, because this is a sort of foundation series, rather than giving a traditional research talk, I'm going to be trying to give you a sense of how I think about statistical approaches for the analysis of microbiome data. So we're going to be thinking about sort of high-level tools, and I'll try to bring that back to problems that you might, or challenges or questions that you might have with a particular case study surrounding differential abundance. So with that, let's dive in. So most of the work that my research group does seeks to analyze this question, which we get from everyone all the time. It's how do I rigorously analyze my data? So you've gone to all this trouble to design an experiment, to maybe recruit participants or find a field site, to collect your samples, to extract your DNA, to go through sequencing, to perform bioinformatics, and you end up with this rich data set that's taken you months and maybe years or maybe decades even to collect. And you want to know how to do the absolute best that you possibly can to learn what your data is telling you about microbial ecology. And one of the somewhat dissatisfying answers that we have to give is that how you should be analyzing your data depends on the questions that you have. So this is a fairly woolly and unsatisfying answer, but I'm going to do my best to think about where do we go from here? How do we give you some insights, even though the answer is it depends. So I think of there as being three different statistical paradigms through which we can think about the analysis of microbiome data, or I guess the analysis of any data. And here we're going to be talking about microbiome data. So the first paradigm is called the inferential paradigm. It's called inferential statistics. And the idea of inferential statistics is that you're not interested in the data that you have as much as what your data says about where it came from. So the idea is that your data reflects some greater mechanism. So you're sampling data from some larger population that you'd like to study, but because you can't exhaustively sequence and you can't recruit everyone in the world or every field site imaginable and process all of that data, you have to make do with the sample that you can. That said, you want to learn about the world sort of in a bigger sense. So that's what inferential statistics is trying to do. A totally different paradigm is the prediction paradigm, and this is often fits into sort of machine learning or computational modeling frameworks as well. So the predictive modeling question is not so interested in anything about some superpopulation that you'd like to study. You're just interested in, well, if we saw someone else, we saw some other sample or another field site, I'd like to know what would happen, what would be the characteristics of that sample. So that's what predictive modeling is doing. And of course, it's learning from the data that you have to make that assessment. But that's the predictive lens that you can take. A final approach that you can consider to analyzing your data is a purely exploratory approach. So what you're doing in the exploratory approach is just saying, I'm not going to try to abstract away from the data that I have. I'm just going to try and see what it's telling me. Are there patterns or consistencies in the quantitative information that I have? Or is there something surprising or unusual? And so there's sort of a different set of rules for exploratory data analysis that I'll touch on in a moment. Most of the presentation that I'm going to give today is focused on the inferential paradigm, which is what I spend a lot of time thinking about. And I'll touch also on exploratory analysis as a contrast, and I'm happy to take questions about any of these paradigms as we go to the Q&A section of this webinar. Okay, so let me give a little bit more details about what the inferential paradigm is thinking about. Inferential statistics is concerned with something called parameters, but in order to define what a parameter is, I need to tell you a little bit more about this paradigm. Okay, so in the statistical inferential paradigm, we think about our samples as being drawn from some population, some mechanism through which data could be generated. And our not so fancy word for that mechanism through which data is generated is called a model. So models are functions or depend on something about the environment, something about the world, something about the universe that are called parameters. So examples of parameters can be numbers. They could be things like microbial abundance, which we'll talk about. They could be sort of evolutionary processes like trees. There are lots of different sorts of parameters, and we'll talk about some of them today. So those parameters, because they're features about the universe or the unknown microbial world, are not known. And our goal in inferential statistics is to try to estimate the values of those parameters from the data that we have. Okay, so that's the high-level picture of inferential statistics. Data comes from a model. Models depend on parameters. We want to learn about those parameters from the data that we have. So a common signpost that what someone's doing is inferential statistics, as opposed to one of the other types of statistics that we've been talking about, is hypothesis testing, or the presence of p-values. So a hypothesis test is a statement about a particular parameter's value. And so I just really want to emphasize that there's no way that the only way to be thinking about hypothesis testing is through this inferential lens. Hypothesis tests are statements about parameters. So that's a dead giveaway. Let me contrast this approach with exploratory statistics, where, as I mentioned earlier, we're not trying to do any abstraction. We're just trying to summarize the data that we have. So there's no idea about mechanism or some universe or parameters. You're just interested in what you're seeing on your desk. And so one of the cool things about the exploratory paradigm is that there are no rules. Okay, so if you want to normalize your data however you want to, if you want to throw away reads in order to have all of your samples have equal sequencing depths, if you want to transform them with additive or centered or other log ratio transformations, you can pick your favorite distance metric between samples and calculate distance matrices, and then you can compute the best two-dimensional representation of your distance matrix using whatever your favorite beta diversity metric is. These approaches are all fine within the exploratory statistics paradigm. But I do really want to emphasize that what I often see is people pursuing what seems to be an exploratory approach, but then finding a pattern and saying, well, I want to know if this is real, with whether this is saying something about the universe as opposed to just my data. And that's where this breaks down, because this exploratory approach, because it doesn't set out with a predetermined parameter that you're interested in testing, it's not compatible with hypothesis testing and p-values. So I have a lot of skepticism about p-values that appear on multidimensional scaling plots, just to give you a concrete example of where this thinking breaks down. Okay, so I've talked a bit about this, this sort of fuzzy ideas about parameters and models. Let's try and get a little bit more concrete by talking about a specific case study. And specifically, I want to be thinking about modeling microbial abundance and what relevant microbial abundance parameters could be. Okay, so let's start by talking about what's a good model for microbial abundances. Okay, so this is my model. My model is that there is some number of a given biological quantity in any environment. Okay, that sounds pretty fuzzy. Let's get specific. So when I say biological quantity, I'm talking about some genetic or biological unit that you define, and that can be dependent on the context in which you're studying your data. So for example, you might be interested in microbial strains or microbial genomes, and then in this case, your biological quantities could be cell counts of genomes. You could also be interested in genes, not respective of their genomic context. And in those cases, the biological quantity that you might be interested in are the number of gene copies in an environment. You could be thinking about sequence variants, for example, 16S amplicon sequence variants. You could be interested in k-mers, you could be interested in transcripts, you could be interested in metabolites. The list goes on and on. Okay, so we've got different biological units, and there's some quantity of each of those units in any environment. Let's give some examples. So, for example, I could have 54,601 copies of this common human skin commensal organism, Staph epidermis, on my right index finger. Okay, that's part of this model for the universe. I could say that there's 874 million copies of this length 11 KEMA circulating in my blood. That's consistent with this model. We could also say that there's 0 transcripts of this particular gene, core RC1 subunit PSSA, on my desk. This is a cyanobacterial gene transcript. Okay, so you would pick which of these sort of contexts you're working in. Are you thinking about organisms? Are you thinking about KEMAs? Are you thinking about transcripts? Are you thinking about sequence variants? And so on. Okay, so let's think about how what that data might look, what that set of parameters might look like. So I'm gonna try to keep this presentation as notation light as possible, but I want to start by thinking about Yij as the true number of our biological unit J in some environment that I'm going to index by I. Okay, so we've got sort of where our samples come from, the environments that our samples come from as rows in this matrix, and then our biological units, whether they're genes or genomes or sequence variants, whatever, are indexed in the columns. Okay, so in addition to this, unknown parameters about microbial abundances, we also have what you might call metadata, so information associated with the environments. So maybe one environment was subjected to some sort of soil amendment. Maybe it was from a patient who had a particular disease diagnosis. It could be from a study site that had high rainfall as opposed to low rainfall, and so on. So I'm using that to just index the colors of my environments here. Okay, so that's sort of a high-level model for microbial abundances. And I want to ask you, if you knew these true microbial abundances, what would you do with them? So there are so many possible things that you could do with them. And maybe I've paused to give you long enough to think of one, and maybe I haven't. But I'll tell you about a couple that you might be interested in. So the first is just the average microbial abundance, the average number of copies of biological unit J, biological unit 4, let's say, across all of our environments. Okay, average microbial abundance seems like a reasonable thing to summarize. Okay, we could also be thinking about parameters associated with the presence or absence of an organism. So we could be thinking about what percentage of environments have the second category, second biological unit present in some abundance. We don't care what it is. We're just interested, is it there or is it not? What percentage of environments is it there in? Another common microbial parameter is species diversity or species richness, biological diversity. So the number of categories that are present in a given environment. Okay, number of different species that are or genomes or genes, whatever it is that's present in an environment. And then the list goes on and on. It can get more complex. I've got one written here, which is the Shannon diversity, where we've defined Shannon diversity as some function of true proportions of abundances. We could be thinking about other alpha diversity indices. We could be thinking about other beta diversity indices. These are all parameters that we could compute if we knew actual microbial abundances. Okay, all possible parameters that are out there. Another source of parameters that are very useful and often used in microbiome studies are parameters that arise through regression models. And so I've talked about sort of environment-specific or category or biological unit-specific parameters on the previous slide, but now let's think about how might we incorporate that information about environment types, where did our samples come from, into a parameter. Okay, so this allows us to make contrasts or comparisons between different microbial environments. Okay, so we could be interested in, instead of asking questions about the average microbial abundance, we could be asking questions about how does that average vary across environment types. And so what I've written here is some regression model. Here I'm looking at the log of the expected value or the mean abundance of the ith category as some linear combination of information about our environment types. So it could just be, I could have these xi's as just being, 1 for our high rainfall environment and 0 for our low rainfall environment. And then we've got a different mean for our high rainfall and our low rainfall environments. And we can then make statements about, well, how does rainfall, how do the means of microbial abundances differ across environments that have different levels of rainfall. Okay, so in this case, our parameters beta are the parameters of this regression model, can be interpreted as fold differences in the average abundance of a microbial category, let's say the jth one, across levels of the kth covariate. Okay, so we're thinking about the exponentiated version of beta kj, and therefore the fold difference in the average abundance of that jth category across the levels of the kth covariate. And I've written it here in grayscale if you're accustomed to thinking about these things in mathematical notation. It's really just a ratio of means when we're comparing groups that are similar in all other environment information, but differ by one level in this kth covariate. Okay, that's another source of parameters in microbiome science. So as we've talked about, parameters are associated with the environment or the population or microbial abundances, which are all unobservable. Okay, so a really hard fact that I have to deal with as a statistical methods developer is that we don't observe microbial abundances. What we have, and for the purposes of today, I'll focus on high-throughput sequencing data. What we have are high-throughput sequencing representations of those environments. Okay, so we go out there and let's say we're doing amplicon sequencing, and in that case, I can count the number of reads I observe for a given sequence variant J in this ith sample, which was taken from environment I, from high-throughput sequencing. I could also be thinking about the depth of coverage that align to reference genome J in sample I, which similarly reflects environment I, from shotgun sequencing, for example. Okay, so I really want to take this point to acknowledge that there is a big difference between what's out there in the environment, these YIs, and the data that we get from high-throughput sequencing, which I'm calling the WIs. Okay, so you'll distinguish the WIs and the YIs because I've got these nice, happy faces associated with the YIs because we'd love to know them, and these sad faces associated with the WIJs because this is the data that we have. And the reality is that as useful as this data is, it is a distortion of true microbial abundances. So these WIs are a distortion of the true microbial abundances. That's really important for what I'll be talking about for the rest of this seminar. So in what way are they a distortion? So I could give a whole lecture on this, but I'll try to summarize some of our research as efficiently as possible. The way that I think about how we get our data, our Ws, from true microbial abundances is that we take our microbial abundance table and we sequence each sample to some potentially differing sequencing depth. Okay, so there's some sampling intensity that's sample specific or environment specific, and I'm going to let those values be denoted by CI. Okay, so we're sequencing some samples more deeply than others. We have this information CI that indexes how deeply we sequence that sample. Furthermore, and this is something that I think hasn't really been connected with statistical analysis of microbiome data, not all of our microbial categories are equally easy to detect. And so in addition to these varying sample-specific intensities, we also have these detection effects associated with each of our different biological units. So every one of these columns has this multiplicative distortion associated with how easy that unit is to sample. So for example, we know that cell wall type interacts a great deal with DNA extraction efficiency, and we are more easily able to pull DNA out of certain cell types, certain bacterial strains and cell wall types than others. So that's just one example. People have talked a lot about GC bias. People have talked about amplification efficiency. There are many different factors that contribute to these detectability effects, but we really need to think about them when we think about how our data is a distortion of what we'd like to know. So in addition to these distortions, sample-specific and category-specific, we also have just random noise generated added on top of that. And this combination of original data with sample-specific distortions, category-specific distortions, and then noise leads us from our true abundances to our observed microbial abundances, these WIJs. So that's really my model for how I'm thinking about our data. Okay. So then the question is, well, I would like to learn about microbial abundances, but I don't get microbial abundances. I just have this noisy and distorted data. So then the question is, well, can I learn what I would like to know about true microbial abundances from this noisy, distorted data? And the answer is, it depends. It depends on what parameter you're interested in learning about. Some parameters you can learn about in the presence of this distortion and noise, and some parameters you can't. You cannot pull out the biology and the sampling effects from each other. Okay, so let me focus on one parameter that you can estimate in the presence of this noise and distortion. So I presented earlier this regression model for the log mean, the log of the average abundances as some function of our environmental type data. But of course, we don't have the W's, so we can't find or estimate the betas. We only have the W's. And so then the question is, can we estimate these betas, these regression parameters from our noisy data W? And the answer is yes. We've just released a tool to do this. It's called RADEMU, and I'll share a link to the preprint towards the end of this talk. I'm really excited about this method, so I'm going to tell you just a little bit about how it works and what it's trying to do. So the sort of one slide version of the hour-long talk on RADEMU is that we're going to combine those two models that I've introduced so far. So the first is that if we take away the noise component, our average observed data, observed number of, let's say, sequencing depth aligned to genome J in environment sample I, is the product of my sample-specific detection effects, my category-specific detectability effects, and these true unknown microbial abundances. And then the other thing that we have is that we're interested in the log mean of these microbial abundances as this regression function that depends on only my environmental data. So we can combine those together to give us a model for the log mean of our Wij as some function that combines parameters from both of these models, and I haven't gone through all of the details here, I fully acknowledge that. And then what RADEMU does is it says, okay, which of these parameters of this log mean W model can we learn about? And it turns out that we are able to estimate these across covariate, across sample type contrasts, which are these betas, and specifically the beta 1Js, the beta 2Js, all the way up to the beta PJs, and that's true for every single one of our biological units. Okay, so you can you can use this data that you have to learn about microbial abundances that you don't observe, which I think is pretty cool. Okay, so let me give you an example of how what you can do with this tool. Specifically in the preprint that we released, we used RADEMU to identify differentially abundant strains in colorectal cancer, in the gut microbiomes of people with colorectal cancer, compared to folks who are otherwise similar but do not have colorectal cancer. I'll give some, I'll make clear in a moment what I mean by otherwise similar. Okay, so to do this, we use this incredible data set. This data set was an international collaboration, and I believe this work was led by some incredible folks at the EMBL, who I'm really grateful to. And one of the things I'm especially grateful to, not only for a very interesting paper, but also is that they made their data available. So that just is an incredible community resource, and I really want to shout out to Werbel and co-authors for making their data available for this study. So what they did was they performed what's called a meta-analysis of 5 different studies, which each performed shotgun sequencing analysis of cohorts that included both folks with colorectal cancer as well as non-colorectal cancer control folks. Okay, so if we combine all of these studies together, we end up with about 566 samples, so of course more than any individual study could see. And after some of their quality control, we end up with 845 metagenomic strains. So they use the MOTUs package. So they're thought of as metagenomic OTUs, but I just think of them as metagenomic strains. Okay, so I told you earlier that what I'm interested in is identifying microbial strains that are more abundant or less abundant in the gut microbiomes of people with colorectal cancer compared to otherwise similar controls. And so what I'm looking for is when comparing populations that are from the same country and the same study, of the same age, of the same level of adiposity, of the same gender, and had similar information about when their stool sample was collected. I'm interested in comparing those populations when there's only a difference in colorectal cancer disease status. So that's one distinction from how we analyzed this data in comparison to the original manuscript. Those authors were looking at folks who differed in colorectal cancer status, but then only within the same study or country, but not but sort of averaging over potential differences in the distributions of age and adiposity and gender, for example, across these different studies. Okay, so what does this tool RADEMU allow us to do? As I said, it allows us to identify strains that are unusually over or under abundant in colorectal cancer samples compared to non-colorectal cancer controls. So what am I plotting here? I can think of every one of these 845 microbial strains lined up along the x-axis, and these lines corresponding to each microbial strain are centered at my estimate of beta. Okay, so remember, we don't know the true betas. They're only information associated with microbial abundances. We just have our data, so we have to estimate them. That's my job as a statistical methodologist. So these lines are centered at that estimate, and then the width of the line denotes the length of the confidence interval. So sets of possible values of beta that are consistent with the data that we observed. Okay, and so what we have, what I've also done here is color-coded the categories that have a false discovery rate corrected p-value that's small. Okay, so we can think of these as categories that are significantly differentially abundant across these groups that are similar in other categories after adjusting for the multiplicity of tests that we have to do. And if folks are interested in why there's some discordance between confidence intervals intersecting the x-axis as well as these colors, I'd be happy to go into that during question time. Okay, so one of the things that I think is really cool about the 20 or so differentially abundant taxa that RADEMU picks up is that it's not only identifying taxa as differentially abundant when they're very common, very prevalent across many samples, and it's not only identifying categories that are differentially abundant if they're very rare. Okay, so we've sort of taken the prevalence, the empirical prevalence of a taxon out of the question of differential abundance, which I think is good because they should be deconvolved. If we were interested in presence-absence, we could definitely do that analysis. But in fact, we're interested here in differential abundance through this regression model, as I defined earlier. Okay, so what I've plotted here is I've just plotted the same y-axis data and the same color data, but instead of just arbitrarily ordering the microbial strains, I'm now plotting on the x-axis the proportion of participants in which each taxon is detected. So on the left-hand side of this plot, you can see all of the estimated effect sizes for microbial strains that were observed, in only 5% or 10% of participants. And on the right-hand side, I can see taxa that were very prevalent, observed in like 80% or more of our participants. Okay, and so we can sort of see that these false discovery rate corrected p-values that are small tend to be across this whole prevalent spectrum. And we tend to see sort of larger effect sizes for more rare organisms and smaller effect sizes for more prevalent organisms, which I think which concords with my intuition at least, and I'm happy to have a discussion about that. Okay, so I've got these nice figures. What about what taxa show up as differentially abundant? So here we're sort of thinking about now we're thinking about sort of hypothesis tests, which I've alluded to but haven't formally introduced. But RADEMU can test evidence against the null hypothesis of beta kj equaling 0, or equivalently that strain J is present in equal abundance in colorectal cancer and non-colorectal cancer samples that are matched on age, study, adiposity, gender, and so on. Okay, so our goal here is to identify microbial strains that are inconsistent with this null hypothesis, so strains for which we would reject this null, and we're going to call those taxa differentially abundant taxa. Okay, so here's a list, here's my list of estimated effect sizes, a subset of all of the significantly differentially abundant taxa that we identified after doing false discovery rate correction. I want to point out, I guess, just two really interesting phenomena. And for folks who are really interested in colorectal cancer specifically, I'm more than happy to give you this list. It's available on our public repository for this data, for this paper. But firstly, Fusobacterium nucleatum shows up towards the top of the list, which I think will surprise no one who's been working in this field for a while. And then another thing that's really interesting, to me at least, is a consistency of estimated effect sizes across different strains of the same genus. Okay, so I can see that RadEmu doesn't know anything about the taxonomy or any information about the labels of each of these microbial strains, but it appears to be estimating effect sizes pretty similar for all of these Clostridia, which I've these pink Clostridia, which I've labeled here in pink, as having exponentiated effect sizes of around 0.4, 0.5. And then similarly, there's this other strain or potential mechanism for Clostridia that's has a larger effect size of around 2, so around twice as the expected abundance of this category is about twice as great in samples with colorectal cancer compared to non-colorectal cancer controls that are matched on other covariates. In contrast, that other one is about half as abundant in non-colorectal cancer gut microbiomes compared to colorectal cancer samples. So there's some evidence of strain switching, which I believe has been brought up in the colorectal cancer setting in the literature. Okay, before I summarize and we go to questions, I just want to tell you a couple of things that I really like about RadEmu, and specifically, I will say that none of the other methods that perform sort of equivalent differential abundant tasks share all of these properties, and that's something that I'm really excited about. So RadEmu is an open source R package. Something that it does is it really clearly connects questions that we'd like to ask about microbial abundances, which we don't get to observe, to what we do observe, which is data about sequencing. It's robust to these differential detection effects, the fact that different categories are differentially well detected, controls type 1 error, so produces valid p-values. Zeros are sparsity, or a lot of categories, biological units that are not observed in many samples, so rare taxa or zeros, are easily dealt with without having to choose a pseudo count. So there's no tuning parameter or robustness that you need to do to what you're going to replace zeros with, which is a property that is not shared by log ratio methods that sort of purport to do the same task. Another thing that it does nicely is it adjusts for the fact that different samples are sequenced with differently deeply. So you don't need to rarefy or equalize or normalize the number of reads across your samples. It's flexible with respect to experimental design. It can work for case control studies. It can work for cross-sectional studies. We have, for folks who are dealing with longitudinal data or co-housed mice, for example, we've got hypothesis test adjustments that can deal with that. And it's very light on the assumptions that it makes. So it provides all of these nice guarantees in very flexible conditions. We're statisticians, we're not graphic designers, so our logo is a little goofy. So if you know anyone who is a graphic designer, I would love to hear from them. Okay, so let me just summarize sort of the key points that I've tried to bring up today. My view is that if we want to use microbiome science to learn about ecology, and we want to learn about evolution, so to make generalizable statements that can be reproduced across studies, we really need to be thinking about what statistical parameters we care about. We need to be thinking about what models are the best way to connect the data that we have to those parameters, and how can we validate the assumptions that those models make, and then how do we develop high quality, open source, rigorous tools to estimate those parameters from data. And if folks are interested in hypothesis testing, how do we ensure that we have valid p-values that apply in a wide range of settings. So to illustrate some of these points, I talked about differential abundance. I brought up this idea of how do we decide, how do we define differential abundance parameters, and I introduced this tool that we recently developed called RadEmu, which is a tool to identify differentially abundant microbial strains in the presence of differential detection. This same philosophy of inferential statistics doesn't only apply to differential abundance, it applies to when our scientific questions are about microbial diversity, when our methods are relevant to presence absence. It could also apply when we're thinking about phylogenomic trees. So I'm happy to generalize this discussion to your favorite parameter if I can. My final request for you is really an invitation, actually. And my invitation is that in any situation where you are reporting p-values, I want you to understand what parameter that p-value is making a statement about, because hypothesis tests are statements about parameters. And I feel that people throw p-values around a lot, even when they're not very clear on what the parameter that's being tested is. So that's my invitation to you going forward. As I mentioned, there's lots of different parameters that you could be interested in. This fold changes method is just one. So RADIMU is a method we're proud of. We have a lot of other tools out there because there are many different parameters that people are interested in. So if you're not interested in fold abundance and you're just interested in presence absence, I'd love to point you to a method called HAPPY. I can provide the reference for that. If you're interested in diversity or species richness, I'll point you to Breakaway and DivNet. If you're interested in Shannon diversity, I'll point you to DivNet and Beta. If you're interested in trees, groves, and so on. So this slide will be available to you if you'd like to go back and check out some of these tools later. So if we haven't met before, and even if we have, I'm Amy Willis. I'm the principal investigator of the StatDiv lab, the Statistical Diversity Lab. We work on what we believe to be the most critical methodological needs in microbiome science and the most serious shortcomings of existing analysis tools. And something that we're also really passionate about are outreach and education and producing high quality software and collaboration. So that outreach part is one of the reasons I'm so excited to be presenting in this workshop today. So I'll thank the organizers again for having me. My thinking on these problems has been informed by a number of incredible collaborators. David Clausen is a graduate of my department, and our preprint estimating fold changes from partially observed outcomes with applications in microbial metagenomics is the RADIMU method that I spoke about today. A lot of my thinking on this work was also shaped by a collaboration with Mike McLaren and Ben Callahan, and that works from this paper, Consistent and Correctable Bias in Metagenomic Sequencing Experiments. This work and all of my work is supported by the National Institute for General Medical Sciences, and I'm very grateful to them for support. With that, I sincerely welcome your questions. I look forward to a lively Q&A, and thank you so much for coming to this webinar.
Okay, so I'll start with the first question. You highlighted how you report p-value, so that's our very first question. Can you please give an example of what you mean by p-values in multidimensional data?
Yeah, great question. So there's nothing specifically about the dimensionality of the data that you have that prohibits a p-value being reported. So again, p-values are statements about statistical parameters. And so you can define whatever statistical parameter is relevant to your multidimensional data. So in the case study that I talked about, I was interested in defining the statistical parameter that we're interested in as these fold differences in microbial abundances for a given taxon. So expected abundance of Clostridium in colorectal cancer gut microbiomes compared to the expected mean abundance of Clostridium in non-colorectal cancer microbiomes. Okay, and then I have that for Calibacterium, and I have that for Fusobacterium, and so on. So those are the parameters that I define. Even though my data is multidimensional, I can test each of those hypotheses, generate a p-value for them, and from there, if I'm interested in making a claim about false discovery rate across this set, I can do that. So I hope that answers your question, but I just want to pull apart this idea of dimensionality of data and p-values. P-values are statements about parameters, which can be defined for data of any dimension.
Thank you. What statistical methods and R packages do you recommend for longitudinal microbiome data analysis?
Yeah, great question. And so I think when people talk about longitudinal data analysis, they're thinking about lots of different things. So as always, the statistical method or the R package that I would recommend would depend heavily on what parameter you're interested in testing. So most regression type parameters that you'd be interested in microbiome science can be generalized to answer longitudinal questions. So maybe people are interested in changes over time, or maybe people are interested in changes over time across groups, or maybe people are interested in the presence or absence of a microbial category over time. So I guess I just want to emphasize that, questions like experimental design and how you deal with the experimental design, I think are secondary to the question of what is the the parameter, what is the question you're trying to ask? Is it about abundance? Is it about presence absence? Is it about evolution? Is it about diversity? And so I feel like we need to talk more about specific parameters before we can talk about tools for longitudinal microbiome data analysis. But yeah, I hope that gives some thinking on a very long discussion that we could have about longitudinal data.
Can you give more examples of parameters we can't estimate from microbial abundances?
Yeah, this is a great question, and I really appreciate you bringing it up. Where I'm coming from with this, okay, so let me give you the most efficient version that I can. So proportions and changes in the proportions of abundances from a specific biological unit are something that cannot be estimated from high-throughput sequencing data in the absence of careful calibration controls. Okay, so what do I mean by that? The challenge is that because different categories are detected unequally well by high-throughput sequencing, it is impossible to pull apart changes in proportions from differences in the detectability of categories. So we go into this question in a fair bit of detail in a paper that I'll just highlight from this previous slide, which is implications of taxonomic bias for microbial differential abundance analysis. So just to say that again, proportions and differences in microbial proportions cannot be estimated from high-throughput sequencing of microbiome data in the absence of control data, which is hard to assemble and hard to collect. The reason for this is that different taxa are detected unequally well, and we go into this issue in a lot of detail in this implications of taxonomic bias preprint, which I hope you'll find helpful.
Thank you. Why do you choose log normalization as your information that you feel is significant for detection of your data? What does this gain or lose in your estimation?
Yeah, great question. So let me do my best to make an educated guess about what you're thinking about here. So my interpretation of this question is that in this model where I'm talking about log mean abundances as some regression function, I think the question is about why is there a log here? Why are we talking about log mean as opposed to just mean abundances? And it's a great question. So let me firstly say that we don't have to be interested in log means or these fold differences. That's just one way, one statistical parameter that we could care about with microbiome data. So on the other hand, there are two practical considerations. So the first is that we can't estimate differences in mean microbial abundances from high-throughput sequencing data. So in the absence of this log, we can't estimate the resulting regression parameters from high-throughput sequencing data. If we had like high-quality qPCR data or ddPCR, droplet digital PCR data, we could fit a mean abundance model. We absolutely could estimate those parameters, but that would require a bunch of category-specific qPCRs, which I guess I would assume people are not willing to do. Maybe we can do this on a small number of categories, but not on the thousands of categories that we can pick up using high-throughput sequencing. So that log mean comes up in order to enable us to estimate the betas in this model. So there's some technical associated with sequencing, and there's some sort of mathematical reasons for that log. And I'll just say that the interpretation is connected, that choice of log impacts the interpretation of the parameter that's estimated. So here, the betas that we get from this model have to be interpreted as fold differences in averages abundances, or the exponentiated betas have to be interpreted as fold differences in average abundances. So I would say that, yeah, all this, how we define the parameter is connected to how we're interpreting it. And I don't feel that anything is lost. Actually, we gain a lot because it allows us to estimate a parameter that we couldn't otherwise estimate from high-throughput sequencing data.
Great, thank you. Can you use Rimu for eukaryotes too, or is it only for bacteria?
Great question. Yeah, the title of my talk was Microbial Abundances, and absolutely, this is always talk about microbial abundances, nothing specific to bacteria here. You can absolutely apply this to eukaryotic data obtained from high-throughput sequencing. So we could be thinking about eDNA or eDNA sequencing variants? Absolutely, great question. Nothing specific to bacteria here.
Could you explain in brief the CI and FDR discordance?
Yeah, great question. Yeah, thanks so much for bringing this up. So this is a part, I guess to explain this, I have to get a little technical. I'll do my best to keep it as broadly accessible as possible. So, so one thing you might learn when you take a mathematical statistics class is that there's not one type of hypothesis test, and there's not one type of confidence interval. So typically, the way that we think about confidence intervals are constructed as estimates plus or minus, let's say, 2 multiplied by the standard error in the estimate. Okay, so that's actually called a Wold type estimate, and equivalently, whether that confidence interval contains 0 is equivalent to a Wold type hypothesis test. There are other types of hypothesis tests, including likelihood ratio tests and including score tests. Those 3, Wold, likelihood ratio, and score tests, are the 3 most common hypothesis tests that are out there. And so each of those tests has different properties and different performance and how well they control type 1 error rate, especially in small samples. And so what we did in RAD-EMU is we used 2 types of hypothesis tests with slight modifications. There's also what's called robust Wold tests and robust score tests. So these are equivalent to Wold and equivalent to score tests, but they're more flexible with respect to the mean variance relationship that's assumed in your data. So they make fewer restrictions, and they control, they give you valid p-values under more general settings. So to answer your original question, what I'm plotting, the discordance here arises from the fact that I'm plotting Wold type intervals, but I'm coloring by score type p-values. Okay, so that's the discrepancy here. I prefer score p-values because they produce better error rate control. We've seen that in simulations, and you can check that out in the paper. So I trust the score tests more for inference when we're really caring about these p-values. But it's very hard to construct confidence intervals from score tests. In contrast, it's very easy to construct confidence intervals from Wold tests. So that's why I've plotted the Wold confidence intervals, but colored by the more what I would consider the more reliable score tests. I appreciate that. Yeah, that's discordant. Thank you for your question.
Thank you. How do you think of this method as different than other computational statistics methods such as ALDEX?
Yeah, great question. So there are a lot of differential abundance tools out there. One of the challenges, a few different thoughts in general, and then I'll speak specifically about LDX2 and other compositional data methods, compositional data analysis methods. So firstly, there's this Nearing and co-authors paper that profiled 14 different differential abundance methods, and they found that they all find different things. So this isn't surprising to me because different differential abundance methods are trying to make statements about different parameters. And there's no reason that we would expect statements about different parameters to coincide. So one of the things that I really like about this RAD-EMU approach is that we have a very clearly defined parameter and a really clear interpretation for what that parameter means in terms of microbial abundances. So I guess I would say that there's been a couple of methods that have come up in the last couple of years that have been trying to do differential abundance testing in the presence of differential detection, but trying to do so in the absence of a clearly defined way to connect microbial abundances and high-throughput sequencing data. And I would describe LDX2 as one such method. So it's a tool to do a thing, but it's not really clear what that thing is. So what it's trying to estimate is analogous in many ways to what RAD-EMU is trying to test. But again, because of that lack of clarity between what it's trying to do and the data that we have, it has to do all of these, what I think of as hacks, essentially. So it does, so you have to choose this link function for LDX2, which is suggested to be a centered log ratio transformed method, that's to deal with differential abundance. But then how do you deal with zeros? In order to deal with zeros, they add this Bayesian interpretation that I don't think is particularly intuitive. Many Bayesian approaches are intuitive. That one is not to me. Similarly, a lot of people have been thinking about analyzing the same question, maybe by just fitting a linear regression model or a generalized linear regression model or a generalized linear mixed model on centered log ratio transform data. And in that case, you also need to confront this question of, well, what do you do when for the sample biological unit pairs that were not observed? So, zeros essentially in your abundance table. And for those, you need to choose a pseudo count, and then you have sensitivity to the choice of pseudo count. What you replace the zeros with is called the pseudo count. And so I would say that those are the biggest sort of functional differences between this approach and methods that purport to do a similar task, like LDX2, like, gosh, what was it called? Some funny term that someone came up with for these types of methods. But yeah, compositional data regression tools often involve a bunch of ad hoc choices to deal with zeros, and they don't have clearly defined parameters. So those are things that I would contrast with this approach. So I'll leave this slide up. These are the things that, like I said, this approach, these are good properties that our approach has that I don't think are shared by any other approach.
Thank you. We have 5 more minutes, but we still have many questions left. So I'm now going according to the likes. So the next question I'm reading is, you used a data set where the colorectal cancer was the only covariate, same age, gender, etc. How can your tool be used when there are multiple covariates? What would you advise in that case?
Yeah, great question. So actually, I think there's been a misconception here, because in fact, we did adjust for multiple covariates in our colorectal cancer data analysis. We adjusted for age, gender, adiposity, study, and so on. So actually, what I presented were the parameter estimates associated with colorectal cancer, but you can think of those parameters as being adjusted, or those estimates as being adjusted for all of these other covariates. So in fact, there were, I guess if you think about coding the studies as multi-level categories, there were 11 or 12 covariates that we did adjust for. So absolutely, this is a regression model that allows you to adjust for other covariates in your model. So they could be confounders, they could be effect modifiers, they could be flexible functional forms like splines or polynomial terms, interactions, whatever you want. So yeah, Rimu is a adjusted regression approach.
Thank you. What is the difference between DSeq and Rimu? I love that you consider the sparsity and that you don't need to adjust for library size or rarefy.
Yeah, great. Thanks so much. I'm glad you're excited about some of the same things that I'm excited about. So DSeq is also, maybe in practice, I would say a big difference is that DSeq is not robust to differential detection. So what does that mean? It means that DSeq is a method that that sort of can't pull apart the impact of taxa being unequally well detected from the abundances of the taxa. So I would say we only care about the abundances. Anything else is a statement about our sequencing process, and we don't want to tie that up into our statements about biology. So DSeq has filled a really important niche and has enabled a lot of really interesting science to happen. But I think, and Mike Love, who's someone I correspond with often, has fully acknowledged that microbiome data is not well suited to the assumptions that DSeq makes. And so, yeah, Mike Love being the first author of the DSeq2 manuscript. But yeah, DSeq doesn't allow for differential detection, doesn't perform well in the presence of differential detection, which is a widely accepted phenomenon in microbiome science. Great question.
Okay, thank you. Have you tested this method on gene counts within metagenomes?
Yeah, great question. We're actually playing around with a couple of gene-resolved metagenomics data sets as opposed to the sort of genome-resolved metagenomics that we applied in the paper. One of the challenges is that these, because RAD-EMU is a joint model, it's estimating parameters on all of your categories simultaneously, as opposed to sort of one category at a time, which is what enables the identifiability of some of these sample effects. It is hard to apply to like the mega-sized data sets that it's harder to apply to the massive numbers of genes that we tend to see in metagenomes. So the answer is yes, there are some computational and speed challenges, and we're working on them. Actually, Sarah Techman, who's an outstanding PhD student who I'm working with, is directly confronting this question and has a really elegant solution. So stay tuned for a preprint on that. Thanks for the great question.
Thank you. What kind of statistical analysis can be performed to tackle technical challenges during metadata analysis if you have raw sequences from different platforms?
Yeah, this is a fantastic question. So one of the ways, okay, so yeah, this is such a good question. So one of the ways that you can adjust for the fact, so what is the problem with analyzing data from multiple studies? There are two challenges, one of which can be addressed statistically and one of which can't. The one that can't is that the populations that that your different studies are analyzing may just represent totally different underlying biological phenomena. So there's no reason that I would expect patterns that I would observe in like urban diet settings to be similar to folks who live outside of modern agricultural systems, for example. So just heterogeneous biological effects, totally different parameters is one challenge that sort of has to be addressed epistemologically rather than statistically. The second challenge, especially when we're comparing maybe populations that we would think are more similar and for which there might be some shared structure, for example, folks with colorectal cancer in Seattle and folks with colorectal cancer in London, is that different approaches to sequencing may have different detectabilities. So we may have one protocol that more easily pulls DNA out of gram-positive bacteria, while the other might pull DNA relatively more easily out of gram-negative bacteria. And so in that case, so one of the things that an approach like RAD-EMU can do is by allowing you to adjust for study, so include study or sequencing batch, you could even think of, as a covariate in your regression analysis, we can directly adjust for those different detectabilities across different sequencing platforms. So what kinds of statistical analysis can be performed to tackle technical challenges during meta-analysis? I would say adjusting for study and choosing parameters that are robust to differential detection. Okay, so something like RAD-EMU, or I should say, like, you could certainly use ALDX2, or LOCOM is another one that you could use for this task. But yeah, certainly adjusting for study if studies may have different abilities to detect different categories. Great question.
Thanks, Amy, for delivering a fantastic webinar and answering all those questions. Is there a place where people can reach out to you, to your GitHub page or somewhere, if they have any question?
Yeah, absolutely. Thanks again, AJ, for the invitation. It's been a pleasure to be here. Folks are welcome to reach out to me by email. I'm not the most efficient at responding to emails, but I do try my best. If folks are trying out our software and run into issues or think there's a problem, we're very responsive to issues posted on GitHub. So if you're giving this method a go and you've run into any problems, I have a whole team to help us with that. So yeah, GitHub issues, when you're running into software issues, get responded to quickly. Emails get responded to more slowly, but you are welcome to email me at adwillis@uw.edu. Thank you so much, everyone, for coming.